I think the role of the learned society in science education and development is one which everyone should appreciate. I explained earlier really that when I went to the University of Bath, not many people were interested in discussing research, and it was really during this time that I became seriously interested in learned societies, because they seemed to me to be the last place you could go where you could actually talk about science with other practitioners in an open and constructive way. I'd always been an attendee at meetings of the PhysSoc and also the PharmSoc, but during my time at Bath, I began to take a very serious interest in the Pharmacological Society. In those days, this was a much smaller society than it is today, but we used to have 4 meetings a year, and in those days, pharmacologists used to go to all 4 during the year. One was encouraged to do that as part of one's training. So the result was you knew most of the people who went to the meetings on regular occasions. You may not remember their names, but you could certainly recognise their faces. You knew where they worked, you knew roughly what they worked on. And the other thing was you always tried to put in an abstract at each meeting, and this was another very important aspect of your training, as was of course presenting an oral communication in exactly 10 minutes and fielding questions from some of the top brains in the field. But it was more than that, because these societies provided a sort of a cradle for the development of younger members, and more senior members of the society often acted as mentors for younger members, and would often, for example, telephone them and say, have you seen there's a good job going that would just suit you, or would you like to come to my lab? I've got a demonstration of another bit of equipment. It was very, very friendly and open, and there was a lot of interaction between those two halves of the membership. I'm afraid a lot of that seems to have disappeared now, and there are various reasons for this. Many learned societies have reduced the number of annual meetings, so that's one reason, so you don't get to see people so frequently. The second reason, I think, is that there are so many more meetings to go to these days. For example, when I started out as a young pharmacologist, you went to 4 meetings of the BPS per year, and to IUPHAR once every 3 or 4 years, and that was your quota of meetings. But now there are so many, no one can afford to go to all of them, so you have to be very selective. And I think the important thing about learned society meetings, and probably about all meetings actually, these days anyways, you don't go there necessarily to learn things, because if that's the case, you might as well stay at home in your office and read papers on your PC, or download something off PubMed or something and read it. The reason you go is this exchange of information, ideas, students, techniques, transgenic strains of animals, and it's the human contact. Science at the end of the day is a human activity. Especially these days, I think the days of a lone scientist have probably gone. Maybe not completely true, but certainly largely true. And most science is done in groups these days, and actually often in worldwide consortia. Just look at some of the genetics papers that have come out recently with 100 authors or something. So I think the nature of science has changed. I think the function of meetings has probably changed. And I'm just sad in a way that perhaps so much of the original culture has disappeared or is disappearing. When I became more involved with the BPS, I agreed to take over the role of an officer position. So in those days, the society was organised in a completely different way to the way it is today. It didn't have a permanent headquarters. The officers were distributed around the country. There were no permanent staff, so everything had to be done using the help of your students, your postgrads, your postdocs or postgrads, or else your PA if you have one. So the first job I took over, the first major job I took over, was meeting secretary, and this was a 3-year appointment, which involved organising 4 meetings a year for 3 years. And I had to travel around the country, seeing people, checking up on their arrangements for their meeting. I saw all the abstracts that were produced. I helped sort them into piles. I helped construct the programme, and I vetted the entertainment suggestions and so on. It's quite a demanding job. And in those days, once you were meeting secretary, you automatically went on to serve a further 3 years as general secretary, which was the old name for president, as it's called now. So this was a further job with different responsibilities, including the general strategic direction of the society, and often chairing many meetings, and most crucially of all, reading the social minutes after dinner at every meeting of the society, which again I'm afraid is a nice tradition which has unfortunately gone by the wayside. But these years were, although they were very, very demanding, but in those days it was considered part of your overall obligations as an academic to help run the scientific infrastructure. In fact, everyone was expected to play a role in running the national science infrastructure. And so it was not exceptional to do these jobs. Nowadays it seems to have changed, and everybody seems to be so much more accountable for their time that any outside commitment that doesn't immediately result in grant money flowing in is rather frowned upon in some circles. But it never used to be, and I think the society was all run by volunteers in those days, and it functioned perfectly well.
The BPS is something that, in a sense, I've grown up with, because it was considered to be absolutely a part of being a pharmacologist in all the departments I've ever been in. And so it was, not a matter of if, but when would you become a member? When would you give your first, in those days you had to give at least one communication to become a member, and when would you do that? And in the department at the College of Surgeons, it was a big event, because everybody who was up to give a paper would be drilled, and they would have to give a rehearsal, and people would ask the most unpleasant questions, purposely, saying, okay, you've got to be ready for something way out from nowhere that you've never thought about before, and you've got to be able to handle it somehow. But what you mustn't do is to pretend it doesn't matter. If somebody asks you a question, you have to treat them seriously, even though you have to say, that was a nice question, but I wish I knew the answer. I just have no idea. You can't say, oh, it doesn't matter, because it's rude and it's not scientific. The rehearsals were absolutely important, intrinsic part of the whole business. But training in pharmacology was not just doing the experiment, it was communication. You had to get to communicate, you had to tell people about your results in an understandable way, and then handle the questions in a sensible way. Too often I felt other people would react almost with hostility to a question. You asked them a question and they seemed to believe it had some ulterior motive, when certainly for me very often the matter of sheer ignorance of the subject. In the department of the College of Surgeons, we had this very strong tradition of having foreign postdocs, and Sergio Ferreira, Salvador Mancada, Arnold Herman, and Americans also, of course, but none of them were familiar with the particular experience of giving a paper to the PharmSoc. And of course, for those whose native tongue was not English, it was a double problem. And So we used to rehearse not just what you said, but how you said it and all that sort of stuff. And it was a very serious, undertaking, and a lot of time and effort and preparation went into producing a communication for the BPS. And later on, of course, when you were a little bit better at it, you would start asking questions about other people, you know, about how they were doing their work, what's happening, what would happen if have you tried this, have you tried the other, and so it was. I remember Sergio Ferreira was very much involved in going to meetings, giving papers, participating in the questions, and of course in the bar afterwards, where so much good pharmacology is done and planned and discussed. This was the place that, I mean, BPS meetings were immensely valuable in talking about problems, getting ideas from people, and everybody would put their tuppence worth in. And you did it. It was an important part of your science. It was an important part of your science. It wasn't an add-on. It was an important part of the science that you were doing.
I think as we move through life, there are obviously key people who are important to us in our development, who nurture us, who take us under their wing, who basically give their knowledge freely with regard to what we're trying to achieve as our personal goal in life. Certainly, the British Pharmacological Society has played a tremendous role for me in meeting many famous pharmacologists, scientists who have basically given so much of their lives to developing new drugs. Sir James Black being one of them, who was a tremendous influence on my life and how the development of beta blockers, which my wife is actually taking now. So people like Sir James Black, people, the management at ICI, people like Barry Cox, Mick Turnbull, then I move into the British Pharmacological Society, Norman Bowery, Rod Flower, Ray Hill, the staff, so many good people with regard to their passion for the science, their passion for helping young people, bringing young people along in the science. And I think that's the thing that I've always tried to bring back to my life. As a young pharmacologist, I found the BPS meetings so important in my career and my life, in both understanding the science, understanding drug development, understanding how scientists think. Many eminent scientists of the day used to sit and ask questions. Now, as I've moved on in my career, I'm asking those questions with regard to young scientists. And to see young scientists at pharmacology meetings and to see their enthusiasm and adopting many of these new techniques to advance neuropharmacology or other aspects of pharmacology is just amazing. And I think it's a great opportunity, as it always has been, for young scientists within the British Pharmacological Society or other learned societies to move forward with their careers and gain the knowledge that people like myself had gained over many years. One of the things I would say to a young scientist coming through with whatever learned society is always challenge the dogma. Because here you are standing up in front of your peer group, basically explaining your experiment. You know that experiment better than anyone. You know the data better than anyone. So therefore, I think it's extremely important, and never be put off by the audience or people around you. Explain it, present the data, and answer the questions. One of my first presentations was in Dublin many years ago, when the chairman had fallen asleep because he'd been at the official dinner the night before and perhaps had drunken a little bit too much wine. He'd fallen asleep in my presentation. I'd finished my presentation and I had to turn to him and wake him up. I was looking around the audience, which was very few, because again it was a Friday afternoon, one of the last speakers of the day, and there was only one person in the audience who put their hand up to ask a question, and that was a professor from Edinburgh. And he asked the question, and I was so pleased, I was so excited, and I gave him an answer which he accepted. So that was my first presentation to the Pharmacological Society, one that I will always remember and always cherish. And this is what you have to look forward to in the future. Hopefully you'll have a few more questions and a chairman who doesn't fall asleep.
So eventually he would get me to prepare a talk, 10-minute talk for the British Pharmacology Society, and I would prepare the talk. I would start off and I'd get about 3 words through, and he'd say, no, what do you want to try and say? We'd go through that. So what do you want to say is this. So that's your first line, learn your first line, get accustomed to where you are. So right, you've learnt your first line. I'd start my first line, I'd probably get through about 3/4 of the way. No, stop that. You should have done that. Go back and do it again. I'd get through the first line, I'd be on the second sentence. No, you've got that wrong. Go back, do it again. Third line, go back, do it again. Go. Stop waving your arms about, you're distracting me. Hold your arms like this. Go back and do it again. No, use your pointer properly. Point on, off, like that. Go back, do it again. But to cut it short, and it wasn't short, we would be rehearsing that 10-minute talk for the vast proportion of that day, by which time I knew it worked perfectly. I knew where to wave my arms and when not to wave them. I knew when to use the pointer. I'd learned my first sentence. I'd learned to say, Mr. Chairman, ladies and gentlemen. I'd learned to say thank you. I'd learned everything. And it took most of the day on the first one. The subsequent ones we did were less, but we still had to rehearse it. And I still do the same to my students.
I've just come from the latest British Pharmacological Society meeting, where we've seen our membership approach 4,000. We have a lot of young people. It suggests to me that pharmacology is going to have a big place in UK science for a long time to come, partly because I think now we are trying to make sure we interact with other societies as much as possible. We're holding more joint meetings with the British Association for Psychopharmacology, that I've also been much involved with the Physiological Society, with the American Society. So I think going back to John Gadham's comment that the pharmacologist is a jack of all trade and a master of none, you know, we're following his ideas from 40, 50 years ago and providing a pharmacological approach to everyone's science, and that has to be good. British Pharmacological Society has always been a major part of my life. I joined in 1973 when it was very much a career step, that after you got your PhD and you'd published a little, you would be expected to join the society, and it was the main to use again a current technique, networking opportunity you had. Could also be quite frightening because you had to present orally. There weren't such things as posters, and people would ask you questions, and since the abstracts were going to be published by the journal, would correct your abstract and make sure that it was correct even down to the last comma sometimes. And I was fortunate, I got involved in helping to organise the 1981 meeting, the 50th anniversary meeting, and I think that led on to me being offered the position as meeting secretary, and that in turn you were expected to move on to becoming the general secretary, which is now called president. I rejoice in the figure, in the title of President Emeritus. They made it up to us by only being called a secretary. Something I enjoyed enormously because I met huge numbers of people outside my own area of interest, and got to know a large number of pharmacologists in the country. And recently went back as a member of council and a trustee because I still enjoy the society and felt it would be fun to be involved at least a little in decision making. But the society has changed enormously. It was a society primarily for giving abstracts and oral and poster presentations 4 times a year, which was a number of meetings I used to have to organise, because it took a long time to publish a paper in the 1980s, and now from acceptance to submission to acceptance to publication you can get something out in a couple of months. In those days it would sometimes take a year, so people wanted to establish primacy of discovery by presenting to the BPS. My very first oral presentation was actually to the Biochemical Society, who met at the Institute of Psychiatry down in Denmark Hill, and at the end I was asked a question about the levels of serotonin metabolites in the CSF. I of course was working just on rats, and said that no, I hadn't done anything, but there had been some really interesting work going on by Ashcroft and Eccleston in Edinburgh studying that very thing. And after I had finished, the person asking the question saying thank you very much, very politely, I went off to see my supervisor, who said, oh, that was a nice presentation, and you know who asked that question? No, I answered. He said, that was George Ashcroft, and I was fairly mortified, but Joe pointed out that all scientists like their own work being quoted back at them. In 1987, Paul Van Hoot came to England to help set up the Serotonin Club. He decided that rather like the Catecholamine Club and the Histamine Club, there should be a club of people interested in serotonin, which is really rather late when one thinks about it. And we met, I think, in the lecture theatre. Anyone was interested of the School of Pharmacy, and he gave a talk about what he wanted to do and how he wanted to set it up. And then I have to say, as far as I was concerned, totally out of the blue, he said, well, Richard Grigg could be the one of the British officers. So yes, thank you. And I then moved on to being vice president and president. Wasn't sure because I was right in the middle of my activities with Astra in those days, so I'm not sure that I was as active as I ought to have been as president. So when the job as treasurer secretary came up, I said yes, I'd do that, and I did that then for about 10 or 12 years, I think, which was very enjoyable, because I had to write a newsletter and help him get involved in organising meetings. We always ran a meeting at Christmas time as a sort of offshoot of the British Pharmacological Society meeting, because a lot of people, from Pat Humphrey to Danny Hoyer, there was a very strong serotonin research in the UK and Europe. John Fozzard is another that should be mentioned, and so we always had a lecture and a dinner on one of the quieter evenings of the BPS meetings. Enormous fun. Recently, I asked actually when I was secretary whether we ought to change the name because Serotonin Club sounded a bit informal when we were trying to get money to help support the meetings, and everyone, including Paul Van Hoot, said no, they liked the name. A few years ago, about 3 or 4 years ago, almost led by Paul, I might say, a move was made to change the name, and it's now officially called the International Society for Serotonin Research, ISSR.
So I think one of the seminal activities in starting up the 5-HT classification studies worldwide was definitely the Heron Island meeting in 1987, I would say. The Serotonin Club had been started by Paul Van Hoot in 1986, and him and I talked a lot about classification, and he was very keen on it and wanted to support it. In fact, Paul found a lot of the money to subsidise the Heron Island meeting, but equally Glaxo Australia put quite a lot in because they knew obviously that we were about to launch sumatriptan in the not too distant future, and so that was the sort of means of the funding. Ewan Mylocrane was the prime mover in organising the meeting and asked me to help, and I said, well, I don't mind as long as it's somewhere decent. And he said, is Heron Island decent? I said, right, you're on, I'm helping you. So off we went, and it was a very exciting meeting. We had a tremendous opportunity to go snorkeling and fishing and all those sorts of things. But I can tell you that during the meeting periods, everybody was in there because it was just one of those meetings where it was buzzing and everybody wanted to know how many 5-HT receptors there were and whether the name they'd given to their pet 5-HT receptor was the right one. And I mean, it was just an amazing meeting, and it was well organised, and I think the climax in terms of classification was the round table discussion where everybody in the conference was involved, and I and Brian Richardson chaired it, and everybody had their sixpenny worth, and in the end Brian and I wrote something up together, and the whole point of that was that I wanted everybody to be involved, everybody to feel part of a scientific fraternity worldwide where they could have their views expressed. And some people were not totally happy with the outcome. I know Michael Gershon, who's a good friend of mine, I love him dearly, but he was very upset these 5-HT1P receptor was not allowed, and it still isn't allowed. So maybe we got it right, I don't know, but we'll have to see. But it was rather funny that evening. Everybody was still very high and got in the bar and people were having a drink, and then somebody decided they wanted to take a photograph of some of the key players, and everybody had to put their hands up with how many receptors they thought there would eventually be. And so we all had to, there was, right, now you go. And I put my hand up, and I only had one finger up. Some people had 10 up. And what I really meant was one thing that meant we found the receptor that's going to provide a drug, and it's sumatriptan. So that was funny. Another thing that wasn't quite so funny was when we were leaving. We had 3 big boats, massive big launches to take us back to catch our planes. We had to cross the Coral Sea, and the one I was in with my wife and other people broke down. There we were in the middle of the Coral Sea. I can't really claim there were white sharks circling around, but it was quite eerie being in this in this boat that was not going to go anywhere. So they had to get lots and lots of small boats to get us back to the island, and then we had to have helicopters to take us to the airport before we missed our planes. So that was all good fun, but a lot of people remember that as well. But it was quite a meeting. I think it was very successful scientifically, and it was great fun, and something I don't think anybody who was there will ever forget.
Another important sort of feature of my career has been my involvement with various organisations. I've been involved with the British Pharmacological Society, the International Club for Serotonin Research, I think it's called today. And also I was involved in the establishment in the monitoring of molecules in neuroscience grouping, which is now an international group. But probably my most important involvement was with the BAP, the British Association for Psychopharmacology, of which I had almost all, I think, the council jobs that were in existence at my time, and I ended up as the president. And after coming off council, I was involved with the governance panel as well. I was the chairman of that for a couple, 3 years, the role of which is to make sure that the association is responding to the needs of its members as well as observing the laws of the association. I think such groupings of scientists are very important because they do allow crosstalk between different types of individuals. So with the British Association of Psychopharmacology, there are both clinical psychiatrists and basic scientists, and their meetings give a great opportunity for a good exchange of views and ideas which can improve the translational value of the research done by both sides. And I think that's one of the great roles of any organisation, I think, to allow good crosstalk, and that can lead to new research ideas, new research projects.
Yes, conferences are really important. I'm very grateful to at least 2 English societies, the Endocrine Society, Society for Endocrinology I should say, and the Society for Reproduction and Fertility, for the opportunities that they have given me over our endocrinology work in sheep. Since these very august old societies were formed, we have set up something called the Clock Club in England, which is a society with no officers. It has no agendas, it has no minutes, it has no records, and it works really well because at one meeting it's decided who's going to do the next one. They get the sponsorship, get the students, and the next one is in Surrey very shortly. So that's been important. But probably, in the American Rhythm Society, which I've been a member of for many years, is vastly important. But the one which is closest to my heart is something which started off life, founded by Professor Johannes Arians Kappers and Dr. Paul Pervet of the Amsterdam Brain Research Institute. And it was called in 1977 the European Pineal Study Group. And I've heard it said, oh, it's full of European neuroanatomists, it was said. Well, it possibly was, but it was absolutely groundbreaking for anybody working on this obscure little organ called the pineal, because it put nearly 200 people in contact with each other. We met every 3 years, and it has now developed into the European Biological Rhythms Society, of whom, Russell Foster was president a couple of years ago, I was president of it in the 90s when it was the European Pineal Society, and it has been really nurtured by Professor Paul Perret from Strasbourg, who was one of the founders, and an extraordinarily energetic and enthusiastic man for keeping people together and enjoying, with enthusiasm, each other's research.
After my rather bruising experience with unemployment, shared by very many of my colleagues, with a group of other senior registrars, we began to do some research into manpower issues in respiratory medicine. We presented this data to the British Thoracic Society, who were very, very uninterested in it and told us this was not the proper business of the society. Saved by Sir John Batten, who telephoned me, and I stood to attention at the other end of the phone. He said, "This is really quite important, isn't it?" I said, "Yes, it is, Sir John." And the BTS set up a manpower committee. And that, I think, was the genesis of the best manpower planning of any specialty. And a spin-off from that was also that the BTS began to get very, very involved in medical education, with postgraduate respiratory education. It's a real powerhouse. Unfortunately, the British Lung Foundation, set up by Sir Malcolm Green and others to raise funds for lung research, came along at about the same time the BTS was getting into gear, and really got out and engaged with the public and with politicians, saying respiratory medicine is a major, major problem and it's important. And I think the two have worked really very well side by side. They've got different functions, except that both of them have resulted in an improvement in the standard of care for people with lung disease in this country. The livery companies of the City of London were set up in the 12th century onwards as trade guilds, providing education and standard setting and support, and they survive. And this Society of Apothecaries is 400 years old in 2017, and was the forerunner not just of pharmacists of this country, but also the genesis of the general medical practitioner. Unlike many livery companies, the Apothecaries has retained its close links with what it originally did and remains as a postgraduate, standard setting and examining body. So it's got a function, rather than just being a dining club. And I became an apothecary about 30 or so years ago because a friend from St Thomas's was a member and we were encouraged to join. And it's probably 15 years or so ago back that I became much more actively involved. I joined the Court of the Apothecaries, which is the governing body, and then in 2014, 2015 I was the Master of the Apothecaries, about the 401st Master, even though we're not quite 400 years old yet. Some of our Masters haven't lasted the full year when they fell out with the King and things like that. But it was a terrific year, which was meeting different people, attending some wonderful occasions like the Magna Carta celebrations at Runnymede and so on, which is just terrific, but also still getting involved with education and training, but in a different environment from the traditional university involvement with education and training. It was a great year, and the Apothecaries, I think, is a terrific and relevant institution.
Well, my story here for this career path that I'm most proud of began in about 1976, living in Vancouver, participating in the Western Society for Pediatric Research meetings in Carmel, California, because the birth defects focus of attention at that meeting was there in Carmel every year. So after these sessions, we'd meet at Clint Eastwood's bar called the Hog's Breath for an after, session beer or two. And as I was leaving to fly home, I noticed there was a Time magazine 2 years old lying on the desk. I thought, nobody's going to miss this. I'll read it on the plane. So I got on the plane, had my dinner, went through it, and here was this half-page story of this 40-year-old woman who had recently buried her brother to the, quote, family disease, and she decided that she was going to do something about it. So she wrote to the National Genetics Foundation asking for assistance, and they sent a neurologist, Roger Rosenberg, and Bill Nyan, a medical geneticist, to interview her, which resulted in a family reunion of some 120 of her family members in Oakland, California, in a park where they had this family room come clinic. And they were now part of what was called the International Joseph Disease Foundation. So I'm interested in neurogenetics. I'll tear this out and take it back, and I stuck it in my basket and under J for Joseph. I forgot about it. And about 2 years later, my department head, who was a PhD, asked me to see this young woman because it involved a physical examination, which she wasn't able to do. So this is a 22-year-old young woman. She's been married. She has a 2-year-old son, and she's in for genetic counseling with her father. And I wondered, you know, why is she coming in now if she was worried 2 years ago? Why wasn't she here? Well, it turns out in the interval, her father had come down with the family disease. And so I said, well, she said, well, this goes back for 10 generations. I said, what do they call it? And she said, Joseph's disease. And over and plucked out J for Joseph, and oh, that's autosomal dominant. I can tell you what's going on. Thanks, end of story. But I thought I'd just see if the people in the foundation knew this family in Canada. Oh, yes, we know this family. You know, not them, but the extended families are large Portuguese families. And how would you like to be on our medical advisory board? I got one patient. I can't. Okay, I'll be on your medical advisory board. So that's fine. I never saw another patient. Another year went by, and I got a phone call from the foundation. We're having a meeting in Lisbon, and we want you to come and participate in the meeting and talk about your research. I don't do research in this area at all. I can't go to Lisbon and take your money for that. Oh no, I really would feel badly about this. So the next thing you know, I get a call from Roger Rosenberg, the neurologist, who's now the chairman or the president of the American Academy of Neurology, is on the line, you know. So we chatted a bit, and he convinced me that I really should come as a medical geneticist, because there weren't too many involved, and just sit and listen and make notes. And at the end of the 3 days, you know, do this particular, what we did, what we saw, wrap-up seminar. So I said, sure, I could do that, and off I went. Well, before I left, I went to another meeting, and on the train, I was reading the issue of the American Journal of Medical Genetics when they were publishing the first article on RFLPs. And I read this, and I said, isn't that interesting? You know, I just filed it away. Got to Lisbon and said, after the 3 days, you've got 200 3-generation families, perfect for linkage analysis. I think you should, you know, seriously go after doing this gene. And they thought it was a great idea, and we wrote it up, and that was the end of the story.
Right, well, the Radical Statistics Group started in late 1974, and it was founded mainly by people who'd been together at LSE in the heady days of 1968. Though those of us working in the health field hadn't been there at the time. I was counting cars in Hertfordshire, and others were studying medicine. And the first thing we wrote was about a document published in 1976, Priorities for Health and Post-Social Services in England. And it was the first time that expenditure on the NHS had been put together in what's now called a programme budget, so it gave you a better idea of how the spending worked by sector. But the argument in the booklet was, at a time when the health service budget was beginning to be cut, there would be less priority to spending on acute services and more priority to community services, mental health, mental illness, older people, and prevention. When you actually looked at the numbers, it didn't show this increase in the so-called priority groups, and which we mainly supported, though we weren't so keen on cuts in acute services unless they were shown to be ineffective, which of course lots of them hadn't been tested at the time. We wrote a critique of this publication and wondered what to do with it. One member of the group had a colleague who lived in a squat in West Hampstead with a group of anarchist printers, and they said they would show us how to put it together as a pamphlet, which they duly did, and we published it. And several members of the group decided to write to the BMJ to draw attention to our critique and to the pamphlet. And when the letter came out, I got into hot water, and my boss, London School of Hygiene, Donald Reed, said, "Oh, I'd never get a job again." Looking at who these naughty dissidents were, there were 3 statisticians, David Jones, Martin Bland and me, and we all ended up as professors either of medical statistics or, in my case, something else. And there were 3 medics, 2 of whom ended up with knighthoods. One was Ian Chalmers, founder of the Cochrane Collaboration. The other was Andy Haynes, who became Dean of the School of Hygiene. And the third doctor was Sheila Adam, who became Deputy Chief Medical Officer, a job which she didn't enjoy very much, but she obviously did it well. So I don't think it could be said to have blighted our career.